Hello and welcome to our final installation of Chapter 7. Today we are going to talk about enzymes. Now, we spent a lot of time last time talking about proteins, and enzymes are a type of protein, so now we're just going to dive into the specifics of enzymes. So let's start with some basic vocab. You should absolutely know these things, but if you don't, that's okay. I'm here to help. So there are three main things about enzymes that you should know: the substrate, the enzyme itself, and the active site. The substrate is the reactant in this reaction. Whatever it is that is being affected by the enzyme is called a substrate. The enzyme itself is a globular protein which acts as a catalyst for whatever reaction it is that we're talking about. The polar regions of amino acids attract the substrate and active site of the enzyme, right? They attract the substrate to the active site of the enzyme. So right here on this enzyme, you would have polar amino acids, and the substrate would be attracted to that so that it would actually interact with the enzyme. The active site itself is the region on the surface where the substrate binds and where the actual reaction happens. The enzymes, once the substrate has been locked into the active site, start the catalyzation of whatever the substrate is. The products are released and the enzyme is used again and again and again, okay? They are specific, right? Like this star shape is not going to fit in this active site, and neither is this circle shape, but this arrow shape would absolutely fit in there, okay? So the enzymes are specific to their substrates. They don't let just anything bind with them. They are only able to work with specific substrates. So how does that happen? Well, there's two main ways that we thought this would happen: the lock and key hypothesis and the induced fit model. Now, the lock and key hypothesis is older and has been sort of the accepted way that this worked for many, many years. And that is where they thought that the substrate would match up with the enzyme either structurally or chemically. So structurally, the 3D structure of the active site is specific to the substrate. That's what we kind of talked about on the previous slide, where it would have to be like perfectly aligned, like you couldn't fit a star in a circle shape, right? Those don't match up. And chemically, if they're not chemically attracted to each other, they won't react with each other. So they need to be both attracted and structurally look the same. Okay, lock and key, right? There's a specific key to every lock. That was that hypothesis. But a better explanation is the induced fit model. So this induced fit model is sort of talking about as the substrate approaches the enzyme, it induces a conformational change in the active site. So it changes its shape to fit the substrate. So you can see here that this is like a very rigid, like arrow shape. And the induced fit model is sort of saying like, yeah, this is like sort of the same shape, but it's not quite the same. And so the enzyme is going to change its shape just a little bit to actually fit this substrate and go through the proper reaction. So it's not an exact fit, but they will force the fit to happen as long as it's like pretty close. So activation energy. Enzymes lower the activation energy of a reaction. That's why we love enzymes, because we can do more chemical reactions with less amount of energy. The activation energy is the amount of energy that must be put into a reaction to make it happen. We don't want to waste energy. That's like living 101, right? Nobody wants to waste energy doing things. And an enzyme stresses the bonds in the substrate, which reduces the activation energy required. And you can see that in this graph, right? So the catalyzed reaction, which is what you would call it when you're using an enzyme, look at, that's like, yeah, there's still some energy being used, but significantly less, and you're still getting the products. An uncatalyzed reaction without using an enzyme, you are using at least twice as much energy, which is like baloney. Who wants to use that much energy? And you're still just getting the same products. So of course we want to lower the activation energy, and of course we want to get our products faster. And that's where enzymes come in. Denaturation is when you structurally change an enzyme so that it doesn't work anymore. This can be done in two ways, a change in a pH or a change in temperature. And basically what this is you're just going outside of its comfort zone, but you're going so far outside of its comfort zone that it will never come back. So if a pH of 7 is its comfort zone and you put it in a pH of 2, it's like, later, dude, I'm never gonna work again. This is your fault. Okay, so if the shape changes, the active site also changes. And if the active site is changed too much, it can't function, and it's just a useless protein at this point. For temperature, you can go high temperatures because the extra energy leads to increased vibrations. And when you increase the vibrations in a high temperature, those vibrations actually break the bonds because the molecules are moving so faster and they're colliding more frequently with each other, and they're like hitting each other so hard that the bonds are breaking. And so when you break those bonds, you break the protein shape, actually. But you can increase the temperature to a certain point and increase the rate of reaction, but that's a very fine line because you don't want to go too far because you can break the protein, you can break the enzyme, but if you raise it just a little bit, sometimes you can increase the rate of reaction, which is like good for you if you want to have the enzyme work a little bit more quickly. And then again, pH, we sort of talked about this. Every enzyme has an optimum pH. It just is wherever you are working the best, that's your optimum pH. When you change the pH, you're breaking the hydrogen bonds. When you break the hydrogen bonds, again, you're breaking the protein structure. You can't get it back once you break that structure. It's usually a permanent change. Once those bonds are broken, they're broken for good. And so then you'd have to build a new protein to get that enzyme back. And then substrate concentration. As you increase the substrate, you usually get an increased rate of reaction, right? If you put more into a formula, you're going to get more out of it. That just makes sense, right? But eventually, you're going to reach the point of saturation. And when you reach the point of saturation, that means all active sites are full and working, and you're not actually going to get any additional increase because you would have to have more enzymes to get more out. So the increase in substrate helps to an extent. Eventually, you're going to level off when you reach that point of saturation. Now, one thing that you guys really actually do need to know is the idea of lactose intolerance. So lactose is a milk sugar. People that are lactose intolerant cannot break down lactose in their bodies because they don't produce lactase, which is the enzyme, in sufficient quantities. And so if you don't have lactase, you can't break down lactose into galactose and glucose. And if you can't break it down, your body does not like that, and it makes you really sick because it's too big of a sugar and you can't use it. So it just exists until you get it out of your body. Those that are not lactose intolerant, that means that you have a sufficient amount of lactase, which allows you to break down this milk sugar into the usable forms, galactose and glucose, which is what your body wants. So, how do we fix this? Well, if you don't drink milk, hello, I don't drink milk, I think it's really gross, you can take a lactase supplement or a calcium supplement. If you are really low on calcium and you're not getting any dairy, you can take a calcium supplement. If you love milk so much and you don't want to go without milk, but you actually shouldn't be drinking it because you're lactose intolerant, you can take like Lactaid, which is a lactase supplement, which gives you the enzyme to break down this milk sugar. And it's just produced industrially using a fungus, and it gives you the enzyme without your body actually having to produce the enzyme. Or you drink lactose-free milk, and this is milk that's treated with lactase, and essentially you're digesting the milk sugar that's in the milk before you actually drink it. So it's still going through the process of breaking down lactose into galactose and glucose, but it's just done in the cart, like before it even gets in the carton and before you even take a sip of it. You can make lactose-free milk by adding lactase to milk, or run milk through an apparatus to remove lactose completely, or lactase completely. But it's like, why? If you are super into milk and you don't want to not drink milk, I would just either buy Lactaid or drink lactose-free milk and not worry about you doing it yourself. So let's talk about metabolic pathways. We've talked about this particular picture already, but we're going to talk about this anyways because all of these pathways are chains of enzyme catalyzed reactions. We love a helpful enzyme, okay? The product of one reaction is a reactant in the next. That's what we mean when we say metabolic pathways, right? So when we have clotting factors, these clotting factors cause prothrombin to turn into thrombin, and once you have thrombin, this is the product of this reaction, it becomes the reactant in turning fibrinogen into fibrin, okay? So you can't even start this final reaction without getting the product of the previous reaction, okay? The Krebs cycle, haven't heard that word in a while, and the Calvin cycle in cell respiration and photosynthesis respectively are examples of cyclic metabolic pathways in which things happen in a circle, right? You go through all of these processes and eventually you get back to the beginning. It is a cycle. Now, enzyme inhibition, there's a few ways we're going to talk about enzyme inhibition. There's competitive inhibition and non-competitive. So in competitive inhibition, the inhibitor fits the active site and just prevents the substrate from entering, right? So we have this inhibitor, and it doesn't really fit. It's not going to do any chemical reaction, but it is going to prevent the substrate from joining in. And it says, haha, like, you lose, I win, you can't be here, right? That's what competitive is doing. Non-competitive, though, it doesn't fit into the active site. Instead, it fits into an allosteric site, which is like another area on an enzyme that has like sort of an opening. It's on all enzymes, but it's not usually where it's not where the active site is, and it's not usually where anything happens except inhibition. And so if this inhibitor fits into the allosteric site, it's going to force the enzyme to change its shape, and when it changes its shape, it changes its active site shape, and now the substrate can't fit in it. So this is non-competitive because it's not going in the same spot, so it's not competing with the space, but it is forcing the substrate to not connect with the enzyme. So what happens? In competitive inhibition, the same maximum rate of reaction will occur if more substrate is added, because we've not changed the number of enzymes available. It's just going to be slower because the amount of enzymes that are unaffected by this inhibitor will still be able to do the reaction. There's just not as many as there were before. In non-competitive inhibition, the max rate of reaction is reduced because there's just fewer functional active sites available for reaction. Okay, if all of those active sites change, we can't go through with the reaction. In competitive, we're not changing the enzyme shape, so it will eventually cycle through all of the substrate. Now there's also end product inhibition, and this inhibition prevents a large buildup of products. So once there's enough end product, they will begin binding to that allosteric site on the enzyme, which prevents additional enzyme activity, right? So as we go through, we have this substrate, it's going to the enzyme, and you're going through this pathway, and you get this end product. Eventually, once we get enough of this end product, it's going to bind to this enzyme, and it's going to change the shape of the enzyme's active site, and it's going to prevent this substrate from being able to attach to the active site, which prevents additional end products from being produced. So 3 different types of inhibition: competitive, non-competitive, and end product inhibition. You should be able to differentiate between the 3, how they are different, and what the impact on the end result is. Great job. We are finished with Chapter 7. Super proud of you. I'll see you in class.